Good day, everyone. My name is Anastassia Mena, and today I'll be presenting my undergraduate thesis, which was conducted under the supervision of Dr. Anastassia Mena from Western University. The title of my project is Hemolymph Viscosity Affects Spider Joint Motion During Vibration Perception. So spiders use their legs for vibration perception, and the way that they do this is through cracks in their exoskeleton called slit sensillae. These sensillae bundle together predominantly at the joints to form lyriform organs. Now, lyriform organs are the spider's sensory organs, and they detect strains in the exoskeleton produced by changes in internal hemolymph pressure. Now, throughout spiders' legs, they have a variety of different channels called lacunae through which their hemolymph flows, and I've included a diagram of that on the right side of this slide. Looking at this diagram, we can see that the lacunae are in quite close proximity to the spider's exoskeleton or cuticle, especially on the right-hand side here. Therefore, the mechanical interactions between the fluid, hemolymph, and the structure may affect vibration perception. So in structural mechanics, there are two properties that are of the utmost importance to this project. The first is stiffness, which is an object's ability to resist deformation when it's subjected to an applied force. And the second is damping, which is the attenuation of vibratory motion that is associated with the dissipation of energy. Now I've included this video of a tuning fork to provide a visualization of these two properties. When you strike a tuning fork on a solid surface, for example, it will vibrate at a given natural frequency, or eigenfrequency. Now from this system, we can get an output, which is called a tuning curve, shown on the right, with frequency on the x-axis and amplitude of displacement on the y. Now looking at this example, we can see that there's a variety of different curves, and the amplitude of these curves tells us about damping, where lower amplitudes indicate more damping and higher amplitudes indicate less damping or no damping whatsoever. Turning now to stiffness, if these peaks were to pop up more so to the right at higher frequencies or to the left at lower frequencies, this would tell us that there was increased and decreased stiffness respectively. Now we can imagine that this tuning fork is filled with fluid, since this is the case in the spider's leg. If this is the case, how might temperature affect this system? Now the reason that we need to consider temperature is because spiders are ectotherms. This means that they cannot regulate their body temperature metabolically. Therefore, their fluid properties are subject to change with temperature. Previous literature has found that hemolymph viscosity follows similar temperature-dependent changes as water, especially within this ecologically relevant temperature range of 0 to 55 degrees Celsius for spiders. We can see that as temperature increases, there's a decrease in hemolymph viscosity. And this brings me to my research question. Does hemolymph viscosity affect spider joint motion during vibration perception? And my hypothesis is that changes in viscosity will in fact cause changes in both joint stiffness and damping. So to address this research question, I took a theoretical multiphysics modeling-based approach using a software called COMSOL. Now within COMSOL, there are a variety of different physics modules that users can use to create their 3D models. The most appropriate module for my project was the thermoviscous acoustic shell interaction module. Now the reason for this is because this module computes the mechanical forces imposed on a fluid by a vibrating solid. It computes the resulting fluid motion. It computes the effect of this motion on the solid. And taken together, all of these computations can be used to predict damping and stiffness due to fluid structure interactions. So following module selection, I moved on to actually constructing the model. And to do this, I took inspiration predominantly from a study conducted by Zentner in 2013, and I've included a diagram from this study in the bottom part of this slide. So looking at this diagram, we can see that she's represent the leg segments and the joint. So to represent the leg segments in my model, I actually used two cylinders, and then I added in a sphere to create the bend at the joint. Turning now to the materials and the material properties of the model, which are highlighted in figure A and B, we can see that in figure A, I've highlighted the joint membrane of the spider's leg, and in figure B, I've highlighted the rest of the cuticle. So looking more closely, we can see that the values for the Young's modulus and the thickness differ between these two areas. Young's modulus is a measure of material stiffness, where higher values indicate a stiffer material. So here we can see that the joint has a lower Young's modulus and is thinner than the rest of the spider's leg, which reflects the softer, more easily deformable joint membrane of spiders. I then specified that the entire model was perfectly elastic, so any force applied to the solid would in turn be released. Finally, I specified that the internal bore of this structure was fluid, specifically water, to represent the hemolymph. Turning now to figures C and D, this is where I specified the area over which any force would be applied to the model. In figure C, we can see it highlighted in blue, and in figure D, we can see the direction of the applied force, where the arrows are pointing upwards. And finally, the last thing I wanted to point out with this model is that I specified the fluid could leave the structure at either of these 2 points, reflecting the bidirectionality of the lacunae within spiders' legs. I then moved on to constructing a more geometrically complex model. So here I again took inspiration from Zentner's study in 2013, and we can see that diagram on the top right. So talking about this figure, Zentner kind of realized that a lot of the volume within spiders' legs are occupied by things like the musculature, the nerves, and the arteries. So she simplified the lacunae into 3 cylinders or circles, as you can see in this 2D diagram. In my model, I reflected this by using 3 cylinders to create the model. And here, one thing I want to point out is that the model is exactly identical to the previous model, except for the geometric complexity, reflecting multiple lacunae rather than just a single one as shown on the previous slide. So all the material properties are the same, and the fluid could leave the structure as well at these 2 ports. So using these 2 models, I conducted 3 computations. The first was to obtain the natural frequencies of the model. The second was to determine the temperature-dependent effects on fluid stiffness and damping. And the third was to simulate vibration perception, which I then used to look at temperature-dependent effects on the frequency response. So turning to my first set of results, here we have the 12 natural frequencies of the simple model with the single lacuna and their respective damping ratios. Now, a damping ratio is a dimensionless value that describes the damping of a system, where higher values indicate more damping and lower values indicate less damping. So if the fluid in this model was not contributing at all to damping, we'd see values of 0 for each of these natural frequencies. But clearly that's not the case, as we can see there's some variation from frequency to frequency. One thing I also want to point out in this table is that each of these natural frequencies corresponds to a mode. And a mode is a manner in which a structure vibrates at a given frequency. So now I'll be highlighting modes 1, modes 5, and mode 11. So here we can see a scale for the coloration on these graphs, where green indicates less displacement and purple indicates more displacement. One thing I want to highlight here is that modes 5 and 11 show most displacement at the spider's joint membrane, where those sensory organs are expected to be located. So now looking at the relationship between stiffness and temperature, here, as I mentioned, the shifts in frequency represent shifts in stiffness. So we're looking for any patterns going on with these modes. But as we can see by looking at the graph, there's no real consistent pattern, which tells us that the relationship between temperature and stiffness is quite complex and should be investigated more in future studies. Turning now to damping versus temperature, here we're seeing a bit more of a consistent pattern, especially for modes 5 and 11 represented in orange and gray respectively. We can see that at the lower temperatures of -10 and 20 degrees Celsius, where hemolymph viscosity is expected to be higher, there's actually more damping going on, which tells us that there's more damping at lower temperatures for this model. Getting a closer look, here we're looking at the frequency response curve, where I applied different forces to this model ranging from frequencies of 0 to 200 Hz, shown on the x-axis, and then measured the total displacement of the model. And we can see that 3 peaks were popping up at values of 25 Hz, 75 Hz, and 115 Hz approximately, and they correspond to the mode shapes shown on the slide. The most important thing to take away from this graph is that we're again seeing a damping observed at the lower temperatures of -10 and 20 degrees Celsius, highlighted by the blue and green curves respectively. So we can see some downward shifts going on, especially at the higher frequencies of 75 and 115 Hz. Finally, moving on to the results for the geometrically complex model that I introduced, starting off with figure A, we can see a kind of different pattern popping up here, where as temperature is increasing, there are upward shifts in frequency, indicating increases in stiffness, which is a bit different from the results from the simple model presented previously. So this again alludes to the fact that future research should look into the relationship between stiffness and temperature, since we're seeing some inconsistent results between the two models. Turning now to figure B, here we're seeing some more consistent results with the previous model, where lower temperatures and higher hemolymph viscosities are corresponding to higher damping ratios, and therefore more damping being contributed by the fluid. And finally, figure C shows the mode of this model at this given natural frequency, where we can see again that the joint membrane is being displaced the most, indicated by the purple coloration, which tells us that those lyriform organs are being stimulated. So what does all of this really mean for this spider? Since the most consistent result in this study was that at lower temperatures and higher hemolymph viscosities, we were seeing more damping being contributed by the fluid, this tells us that vibration perception in these spiders may be hindered at lower temperatures. Furthermore, as I pointed out, a lot of this damping was occurring at higher frequencies, so this tells us that spiders might be less able to detect these frequencies when they reside outside of their optimal temperature range. So clearly we can see that temperature plays an important role in affecting the spider's sensory system. Some next steps for this work are to create a more accurate depiction of the spider's leg, accounting for things like variation in geometric dimensions from species to species, or variation in material properties from species to species. Furthermore, future work should again look into the relationship between stiffness and temperature being contributed by that fluid viscosity. Finally, before I wrap up, I just wanted to acknowledge the help of my supervisor, Dr. Anastassia Mena, and Dr. Aaron Brandt from our lab. I couldn't have done this project without them. Furthermore, my advisory committee, Dr. Graham Thompson and Jacqueline Lebenzon, I'd like to thank them for all their help throughout this process. And finally, thank you to my lab mates. I got a lot of feedback from them, and they really helped this project get to where it is now. Thank you for listening. I'll now be taking questions at this time.